Good evening, and welcome to our 17-part series, An Evening with AP Biology. On night one, we're going to focus on macromolecules, and we're going to start with the macromolecule carbohydrates. Most carbohydrates that we're going to talk about here are going to have a base in glucose. Glucose has a molecular formula of C6H12O6, and there's the glucose molecule. Now, glucose can be put together in, shown here, three possible orientations. We can store our extra glucose in what is known as glycogen, and we are going to store that in our liver. And if you notice, it is sort of branched where it's bound right there, and we can store a lot. And when we need to increase our blood sugar, we break down glycogen in our liver. If we were a plant, we would store our glucose in the form of starch. Now, if you see starch and cellulose, they look very, very, very similar. Cellulose is going to be found in the cell walls of plants, and starch will be found in things like potatoes and in the roots. We can break down starch in our digestive system, so we like it. Pasta, potatoes, things like that. But cellulose, we have a little bit of a harder time. We really can't digest that. And it looks pretty similar. In starch, the OH groups are all on the same side. If you look in cellulose, they flip. So that is a major difference between starch and cellulose, all made up of glucose molecules. If we take a look at your carbs are going to contain carbon, hydrogen, and oxygen. I've seen a lot of questions over the past several years, and knowing just these little basics like what elements are found in these compounds is very important. So carbohydrates do not have nitrogen, they do not have phosphorus, they do not have sulfur. Hydrogen and the oxygen in a carb will be in a 2-to-1 ratio. And just happens that the hydrogen and oxygen and glucose are in a 2-to-1 ratio. We can break down starch, as we said, and the glucose can then be used to go on to create some ATP in cellular respiration, which we can do. Single-celled organisms like bacteria, get rid of that extra L there, will also use glucose, but they're going to use glycolysis, and they can do that in the absence of oxygen, called anaerobic respiration. As stated before, we can store glucose in our liver in the form of glycogen, and we can't break down cellulose, which I said. But cows have a diet that has a lot of cellulose in it, and in order for them to basically extract the nutrients, they need a mutualistic relationship in their gut with some microorganisms, and it's mutualistic, it's plus, because the animals like cows, the microorganisms will extract out the nutrients from the cellulose, and the cow gives those microorganisms a place to live. So it is a mutualistic relationship. Moving our story along to proteins. Proteins are composed of building blocks called amino acids. There are around 20 amino acids, and all of them will have the base kind of formula of NCC. On the N side, there'll be 2 hydrogens attached. That's called your amino group. On the other side, there'll be a double bonded oxygen and an OH. That's called your carboxylic acid. Everybody gets a hydrogen attached to the central carbon. And then there's this R group, or this side chain. Now, the side chain could be negative, it could be positive, it could be polar, it could be nonpolar, it could be a lot of things. And that side chain tells a big story on the protein once it's finally all said and done. So proteins have carbon, and they have hydrogen, and they have oxygen, but as I just stated, they have nitrogen as well. So now we've stepped it up. Some even have sulfur. That's going to be a clue for later. Sulfur. These amino acids, when they get joined, once again, this is an amino acid right here. This is an amino acid right here. And we don't know what the name of these amino acids are, is because they only have an R group. They didn't tell us what the name is. But when you put them together, you are going to make a peptide bond, and that peptide bond is going to be right there. And whenever you make one peptide bond, you are going to release an oxygen, a water, in a process known as protein synthesis. Now, these polypeptide chains will go through a series of foldings and interactions and go on to form functioning proteins. Proteins will be assembled with the instructions from the DNA. Amino acids will continue to be added until you reach what's called a stop codon, and we'll talk more about that in protein synthesis. But at this point, the amino acid chain or polypeptide will break free from the ribosome and fold and begin to function. Proteins are very, 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 very specific, and if anything changes, temperature, pH, they're not going to fold right, and if they do not fold right, they're not going to function. Also, if there is any mutation or any error in the DNA, that's going to send the wrong instructions and the wrong protein will be made. They are found everywhere, and they can be structural, like in muscles. They can be found in hormones, neurotransmitters, and enzymes that make or break other molecules. Catalysts, they can be found in the surface of cells as receptors. So they're everywhere. So without them, you'd be in trouble. And that's why when just temperature changes a little bit, if we have a fever just a few degrees higher than our body temperature, that could be it for us, because if the proteins unfold or denature, they're not going to function, and that could not be good for us in the end. Lipids, they're going to be in our, I guess we'll call them our fats family, and they're going to be like the carbs. They're going to contain carbon, and they're going to contain hydrogen, and they're going to contain oxygen. They are not, however, going to have the hydrogen and oxygen in a 2-to-1 ratio. There is going to be many, 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 many hydrogens and only a few oxygens. And I'll show you what that means in a second. They're going to have a glycerol. They may have a phosphate group if they're a phospholipid. They'll have fatty acids. We're going to talk primarily about phospholipids here, and they're going to be what are found in the cell membrane. They're going to have two ends. They're going to have a hydrophilic end, and they're going to have a hydrophobic end. So let's kind of talk about the phospholipid. If we zoom in here, you can see there are two fatty acid chains. That's one fatty acid chain. That's another fatty acid chain. And that double bond is going to make it unsaturated. And the lack of double bonds here is going to make this one saturated. It says it right there. There's our phosphate, and there's our glycerol. Glycerol is a 3-carbon molecule. In this case, it's written sideways. And the phosphate group is in the what's known as hydrophilic head, and the tails are hydrophobic. If we come over to this version, you can see they'll sometimes draw it simple with your polar head or your hydrophilic head and your non-polar hydrophobic tails. And these are going to go on to form the lipid bilayer, which you will see in your cell membrane. Water is not welcome here. Not welcome at all. Okay, now we move our story to nucleic acids. Now, nucleic acids are going to be your DNA and your RNA. We'll focus a lot more on these as we go. But these will contain carbon and hydrogen and oxygen and nitrogen and phosphorus. These are the building blocks, or the building blocks are called nucleotides. And a nucleotide is going to have 3 parts. It's going to have a phosphate group, it's going to have a base, and it's going to have a sugar. If you are DNA, you're going to have the bases adenine, thymine, guanine, and cytosine. If you are RNA, you're going to have adenine, uracil, guanine, and cytosine. Notice no T in RNA. And that will be a huge giveaway, talking about DNA. DNA is also a double-stranded molecule, and what that means is that they're, in essence, just reverses of each other. So everywhere there's a C, it's color-coded right, there will be a G. So therefore your C's will equal your G's, and your A's will equal your T's. That's Chargaff's rule. RNA does not have any rules like that because RNA is only single-stranded, and because it's single-stranded, it's not going to have to worry about any complementary bases, and it's going to have uracil and not thymine. So let's see what we got. I like this little flow chart right here. We know RNA is going to have a ribose sugar. DNA is going to have a deoxyribose sugar. We kind of draw a little nucleotide here, so they'll draw a nucleotide pretty simply. They'll have a little phosphate group up there. They'll have your sugar, which will either be a ribose or a deoxyribose sugar. And there's your base. Depending on DNA or RNA, that base will be, either A, U, G, and C or A, T, G, and C. RNA is going to have uracil, while DNA is going to have thymine. And how are they the same? Well, they're both polymers, so they're both large molecules made up of small building blocks. They are types of nucleic acids, and they are found in all living things. Now, all living things may not have a nucleus, but they will have DNA and RNA. Whenever we build a nucleic acid, which once again it is made up of nucleotides, we will add them to the 3' side. Now you say to yourself, what does that mean? Well, the sugar has an oxygen up there, and it kind of looks like sort of home plate kind of. And that's going to be the 1' carbon, and that's going to be the 2' carbon, and that's going to be the 3' carbon, and that's going to be the 4' carbon, and then the 5' carbon is going to be up there. And when you want to add nucleotides together, you add them this way onto the 3' carbon. You're going to actually form what's called a phosphodiester bond, which is right here, or phosphate diester linker. And when you put those together, you are going to, of course, release water, just like when you build an amino acid. When you build a protein. And let's see. Yep, and this will happen in both DNA and RNA. So hopefully that was a nice intro to macromolecules. Let's review. Carbohydrates, glucose can be put together to form glycogen, cellulose or starch. And we can break down starch if we eat it. We have the digestive enzymes for that. We do not, however, have the digestive enzymes for cellulose, so don't eat grass. If you are a cow, you do have a mutualistic relationship with some microorganisms. Then we move our story to proteins, which are chains of amino acids, and their folding pattern determines their function. And we went on to lipids. We only talked about phospholipids, and they're found in the cell membrane. And they have that kind of hydrophilic head and that hydrophobic tail. They form a lipid bilayer. Then we moved on to nucleic acids, where DNA is double-stranded, where A equals T, G equals C. RNA is single-stranded. RNA can leave the nucleus, which we will find more about in protein synthesis. It is single-stranded, and it contains uracil and not thymine. So hopefully that was helpful, and we look forward to night 2. See you then.